really sorry for the problem, the connection issue that we had. So welcome to this new NSFA webinar. So we will talk today about lymphatic and cardiometabolic diseases. So we'll start now with Véronique Angéli. Véronique, are you here? Yeah.
Thanks, Soraya, for this kind invitation and to organize this webinar on lymphatics and cardiometabolic diseases, and to give me the opportunity to share with you some of the work done in the lab about the role of lymphatic dysfunction in atherosclerosis and cholesterol buildup, and how improving lymphatic function via cholesterol reduction may offer a therapeutic strategy. So since I'm the first one to talk about lymphatics, I will just give you a very brief introduction about the lymphatic system, which is basically consists of a large network of lymphatic vessels with interconnected lymph nodes as shown on this diagram. And so in the peripheral tissue, your initial lymphatics or capillaries will drain the lymph towards like larger lymphatic vessels, also called the collecting vessels, which are covered by smooth muscle cells. And then the lymph will drain into the lymph node and then back to the blood circulation. So lymphatics are, the primary function of lymphatics is indeed to drain fluid and to maintain fluid homeostasis in tissue. And so if lymphatics are obstructed, altered, then as a consequence, you have accumulation of fluid and induction of swelling or edema, or called lymphedema. But lymphatics are also very important in immunity, like by transporting immune cells, antigen, and also resolution of inflammation. But in the lab, we've been very interested in the past decade on the function of lymphatics in lipid transport, and particularly cholesterol. Of course, I don't have to mention too much about cholesterol, but cholesterol is definitely very important for cell membrane formation, the formation of steroid hormones. But of course, if it accumulates in excess, then it can be basically toxic to the cells. So cells have evolved mechanism to basically prevent this excess of cholesterol within the tissue. And one of the mechanisms is the reverse cholesterol transport. So in this process, basically peripheral cells will efflux cholesterol through transporters, and then this cholesterol will be accepted by high density lipoprotein, or HDL, and this HDL cholesterol will be then transported through the blood circulation to the liver, and then for excretion via the feces or reabsorption in the intestine. However, when we start this work at the time, so it was back to 2010, I think, it was unknown how basically cholesterol returned from the peripheral tissue back to the circulation and then towards the liver. And there were interesting reports suggesting that human lymph is very rich in lipoproteins. And so we hypothesized that maybe the missing link in this reverse cholesterol transport might be the lymphatics. So to show whether lymphatics are indeed essential for this reverse cholesterol transport, we established a model where we can track specifically the cholesterol transport in vivo. So we basically loaded macrophages with fluorescent cholesterol, then injected them subcutaneously in the foot pad of the mice, and then follow basically this fluorescent cholesterol from the site of injection into the lymph node, the lymph, plasma, liver, and feces. So as shown here, when we track this fluorescent cholesterol, we can see a progressive clearance from the site of injection, so the foot pad, and this cholesterol will then be drained into the lymph node, and later on we can track in the lymph, plasma, liver, and even the feces. So to investigate whether indeed this reverse cholesterol transport is dependent on proper and functioning lymphatics, we impair lymphatic drainage in these wild-type mice by surgically basically disrupting the lymphatics. So shown here, we disrupt these efferent lymphatics, and that basically definitely block the drainage of the dye. Here, for example, physics to the draining lymph node. So we compare the transport of cholesterol in mice which are functioning lymphatics and those that have basically impaired lymphatic drainage. And you can see that this cholesterol is now accumulating when the lymphatic in the footpad when the lymphatics are basically resected. And as a consequence, basically the transport of cholesterol from the site of injection to the lymph, plasma, liver, and feces was significantly reduced. In fact, we show that lymphatic vessels, like the impairment of lymphatic drainage, like reduced by 80% of this reverse cholesterol transport. In fact, the group of Gwendoline Randolph had similar results, but using another mice of lymphatic deficiency, which is the Chi mice, which lack lymphatic capillaries in the skin. And she showed in these mice that also the reverse cholesterol transport was significantly altered. So in this first part, I just show you like the importance of this lymphatic drainage in basically for an efficient reverse cholesterol transport and basically preventing the accumulation of cholesterol in the tissue. However, it was interesting in the lab, we have also data suggesting that high level of cholesterol in the circulation, so hypercholesterolemia in mice, was associated with compromised lymphatic function. And these were from data from ApoE knockout mice, so these mice that lack apolipoprotein E, and which in fact so develop hypercholesterolemia and also is a model of atherosclerosis. So in these mice, we show indeed that lymphatic transport was reduced, and this leads to tissue swelling, accumulation of cholesterol and lipids. And in fact, we show that this impairment of lymphatic drainage was associated with changes in the lymphatics. For example, initial lymphatics become more dilated, and then the collecting vessels, which are normally wrapped with smooth muscle cells, here we could see a decrease in this smooth muscle cells coverage. And in fact, later on, we show that the pumping activity of these vessels was significantly impaired. So basically, what I've shown here is that there is this bidirectional crosstalk between cholesterol and lymphatics. In one hand, lymphatics are really important to for cholesterol transport and to prevent its excessive accumulation in the tissue. But on the other hand, when you have this excess cholesterol, this can impair lymphatic transport and structure. Of course, this identifying this crosstalk led to more questions, and particularly one question was like, what is the relationship of lymphatic function to atherosclerosis? Because indeed, atherosclerosis, as you know, is a chronic inflammatory disease of arteries characterized by the accumulation of intima cholesterol and immune cells. Interestingly, there were these hypotheses raised by Gérald Lemol, who is a cardiovascular surgeon in the 80s, who basically suggested that lymph stasis might promote the development of intimal atherosclerotic plaque, but there was no direct evidence yet for that. And then also there were several work that show, in fact, that atherosclerosis is associated with the growth of lymphatic vessels, also known as lymphoangiogenesis, particularly in the adventitial layer. So this is the most outer layer of the arterial wall. And in fact, increased adventitial lymphatic densities correlates with intimal thickness, and notably, particularly in advanced plaque. In our hand, we also show that ApoE knockout mice show some lymphogenesis, particularly in the aortic sinus whole mount, where you can see in the ApoE this expansion of the lymphatic network compared to wild-type mice. We could also detect some lymphatic sprouting, and indeed lymphatic vessel number also correlate with the size of atherosclerotic plaque. However, at the time, it was not clear whether this expansion of lymphatics or lymphogenesis was just part of the pathogenesis of atherosclerosis or really an attempt to increase lymphatic drainage. So we decided to really assess the lymphatic transport in the aorta. We decided to focus on the abdominal area because we could detect beautiful lymphatic vessels. And in this assay, basically, we inject like a macromolecule, a dextran, in the adventitia layer and then follow the transport of this macromolecule into the lymphatics down to the draining lymph node, which are the iliac and the renal lymph node up to the thoracic duct. And we can see here indeed that we can detect this fluorescent dye into the draining lymph node. I have a small video here to show how basically this dye after injection in the adventitia layer was uptaken by the lymphatics and then further down, drained further down in the lymph node. So with this assay, we went on to compare basically lymphatic drainage in wild-type mice or ApoE knockout mice. And we show that in these ApoE knockout mice, we had a decreased transport of this tracer into the draining lymph node, so iliac and the renal lymph node. And then we also show that when we treat mice with ezetimibe, so ezetimibe is a drug lowering plasma cholesterol and has been shown to decrease atherosclerosis and hypercholesterolemia. And in this case, you can observe that basically we have an improvement of this lymphatic drainage when we reduce hypercholesterolemia. So here really showing that although we have more lymphatics, but we have an impaired lymphatic drainage. So of course, the following question was like how this lymphatic drainage is important in the plaque development. So to address like how basically lymphatic drainage can like affect the development of atherosclerotic plaque. In ApoE knockout mice, we ligated, basically, we tried to interfere with the lymphatic drainage before, of course, we observe this impaired lymphatic drainage and before they develop atherosclerotic plaque. So we ligate these lymphatic vessels, and this basically block, as shown here, the transport of the molecule into the iliac lymph node. Basically in the iliac lymph node, you see an accumulation of this dye because it's not drained into the more distal lymph node, the renal lymph node. So here we basically compare ApoE mice with effective drainage or with ligated lymphatic vessels. And we first show that there's no alteration in the total plasma cholesterol, but we clearly see an accumulation of the cholesterol within the aorta when we alter lymphatic drainage in these mice. What about the atherosclerotic plaque? In fact, as shown here, when the lymphatics were non-ligated, we didn't see any yet like a plaque, but as soon as we ligate the lymphatics, you can appreciate the formation of this plaque, as shown here by the accumulation of macrophages and that are like uptaking lipids. And this is shown here where you can definitely see an increased plaque area when the lymphatics were ligated in these ApoE knockout mice, as well as more immune cell accumulation was observed also, like as dendritic cells and T cells that are known to usually migrate out from the tissue by the lymphatics. So here really suggesting that lymphatic ligation can promote atherosclerotic plaque development. So we went next, basically, to further, support these results. We assess like how basically lymphatic drainage is important in the regression of atherosclerosis induced by ezetimibe. As I mentioned earlier, ezetimibe is lowering cholesterol and has been shown to induce a regression of atherosclerosis. So here we treated all APOE mice with ezetimibe, but we had a group with intact lymphatics and another group where the lymphatics were ligated. And as shown earlier, like basically this doesn't impact the total plasma cholesterol. However, what was interesting is that compared to APOE mice treated with ezetimibe with intact lymphatics, we could see a basically larger plaque as shown here and by the quantification. So suggesting that the regression of atherosclerosis induced by ezetimibe is really like dependent on the efficient lymphatic drainage. So I've shown you so far that lymphatic drainage is compromised in atherosclerosis despite this lymphoanginesis. So it means that I think we have to be cautious when we basically we observe an increase in lymphatics. It doesn't necessarily mean more, but better. And also here we've shown that basically indeed lymph stasis promote the development of intimal atherosclerotic plaque and adventitial inflammation, which is consistent with some other reports. And then that efficient lymphatic drainage is essential for regression of atherosclerosis. So suggesting that we need to develop strategies to promote lymphatic drainage for treating atherosclerosis and to induce this regression of atherosclerosis. And I think this will probably be covered by Sebo et al. Later on. So here I've shown you, the concept that lymphatics are indeed really important to drain, to transport cholesterol and to prevent its excess accumulation in the tissue. However, all this was done in mice, and to us it was really important to see the relevance of this concept in humans. So how to address this in humans? So we decided to look at condition of non-condition associated with lymphatic insufficiency and to see whether there is indeed in this condition cholesterol deposition. And we focus on secondary lymphedema. So secondary lymphedema is a condition that is occur in cancer patient post-treatment, about in 20-40% of the patient, and this is because during the tumor removal, lymph node sentinel lymph node removal, radiotherapy, this alter basically the lymphatic vessels and basically induce impaired lymphatic drainage, which results in fluid accumulation as shown here in these pictures. But not only this impaired lymphatic function does not only lead to fluid accumulation, but remodeling of the tissue, and particularly adipose tissue, is associated with inflammation and at much later stage severe fibrosis. So we decided, so we collaborated with these plastic surgeons back in Singapore, Prof. Tan Bien Kim, who is a plastic surgeon and manage such a patient. And so he could provide us with full thickness skin biopsies to basically analyze whether there is indeed accumulation of cholesterol. So we had biopsies from healthy and stage 2 and more advanced lymphedema staging. And we exclude obese patients because obesity has been shown to exacerbate lymphedema. So we just investigate in this tissue whether we could observe or not this accumulation of cholesterol, and we particularly focus on free cholesterol, which is the cholesterol that is known that if it's accumulating, it's more toxic. And this free cholesterol can be assessed by a Philippine stain, which clearly specifically identify free cholesterol and not the esterified cholesterol. We found that compared to healthy, we have an accumulation of this free cholesterol in the dermis, but also in the dermal adipose tissue, as shown on these images, but also in this quantitative analysis. And this approach was further confirmed by using mass spectrometry, where we found indeed an accumulation of free cholesterol in lymphedema tissue. And since I showed you earlier that lymphatics are really important to drain cholesterol, we also, I forgot to mention that at later stage, in fact, it was really interesting to find that these cholesterol now accumulate as in the form of foam cells. And as you know, foam cell usually is really characteristic of human, of atherosclerotic plaque. But here in lymphedema tissue, we could also identify these macrophage and fibroblasts that pick up basically these lipids. So as I mentioned earlier, lymphatics are important to transport cholesterol. So we were curious to know whether we could see some specific accumulation around of cholesterol around the lymphatics. And we look at initial lymphatics first, so stained by D240 and Philippine. So you can see that unlike in the healthy lymphatics, we could really in the lymphedema patient, in this initial, we can detect an accumulation of these free cholesterol around the lymphatics. And this was also observed around the collecting lymphatic vessels. It's known that during the disease, the collecting vessels also remodel. For example, the lumen is more narrow. There is an accumulation, proliferation of smooth muscle cells as well as collagen. And in all these different phenotype of collecting vessels, we could also observe some accumulation of cholesterol around the vessels. So this suggests that during when you have lymphatic insufficiency in lymphedema, this leads to this accumulation of cholesterol. So we wonder whether can we target this excessive cholesterol and deposition and to alleviate secondary lymphedema. So of course, the question was how to target this cholesterol. Is there a way we can basically remove this cholesterol from the tissue? And we focus on this molecule called cyclodextrin. So cyclodextrin are cyclic oligosaccharide compounds, and they've been shown to increase basically cholesterol solubility. And in fact, there are several papers showing that it promotes the removal of cholesterol from foam cells. And particularly, it was really interesting this paper by Eikelatz showing that, in fact, the treatment of apoE mice with cyclodextrin promote atherosclerosis regression, and particularly by promoting cholesterol efflux by macrophage and macrophage reprogramming. So we decided to use this cyclodextrin, but first we also had to develop a model of secondary lymphedema to test the effect of cyclodextrin on lymphatic function and swelling. So we established this preclinical model of secondary lymphedema, which really mimic the human lymphedema. So basically, this is induced by resecting the lymphatics in the mouse hind limb, and you can see that this will induce this swelling of the hind limb. And of course, we further confirm that in this model, we also see this cholesterol accumulation as observed in human lymphedema, and this is the quantification. So we use this mouse model of lymphedema and treat some of the mice with cyclodextrin. And although cyclodextrin does not affect the total plasma cholesterol in these mice, we could observe a significant reduction in the accumulation of free cholesterol in this tissue. So then what about the effect on swelling? And we could observe also a significant decrease in the lymphedema. And importantly, this was associated with an improvement of lymphatic clearance, as shown on this graph, where we can see a reduction of the basically fluorescent marker, fluorescent molecule within the peripheral tissue. So really showing that cyclodextrin can alleviate lymphedema, the swelling, and through improving lymphatic function. So of course, what is the mechanism underlying this beneficial effect of cyclodextrin? We look whether this improvement in lymphatics was associated with some changes in the lymphatics, and we could observe some growth of lymphatic vessels, as shown here by the quantification of initial lymphatics in the peripheral tissue after treatment with cyclodextrin. And to further confirm this potential effect of cyclodextrin on the growth of lymphatics, we use another model which helped to look at the regeneration of lymphatics. So in this model, basically, we resect a small section of the lymphatics, and over time, basically, the lymphatics will reconnect. But here we have a group that was treated with vehicle and another group which was treated with cyclodextrin. And we can see that with the treatment with cyclodextrin, we significantly enhance the percentage of reconnection of these lymphatics, suggesting that it helped to regenerate lymphatics. And this basically process is dependent on the signaling of VEGF receptor 2 and 3, which has been shown for a long time to be very important for lymphatic maintenance, but also growth. So basically, in this last part, I've shown you that we like to propose that secondary lymphedema pathophysiology involve this abnormal deposition of cholesterol. And I didn't present you all the data, but we propose that it's really driving some of the changes observed in this tissue during secondary lymphedema. But also we provide evidence that removing this excess tissue cholesterol using cyclodextrin can improve lymphatic drainage and lymphedema. So we think that maybe, in the paper from Ekelatz showing that cyclodextrin induced a regression of atherosclerosis, maybe another mechanism by cyclodextrin might be in fact that it improved lymphatic function and contribute to this regression of atherosclerosis. So with that, I like, of course, to acknowledge the people that were involved in this work, and particularly a postdoc in the lab, Angeline, who has contributed to the majority of the work presented today. Joachim, who worked on the more atherosclerosis, and lately a PhD student, Yuning, and also a technician, Shaza, who worked on the lymphedema project. All are collaborators from Singapore and overseas, and also all the patients participating in the last part, so the lymphedema study and our funding agency. With that, I'd like to thank you for your attention, and we'll be happy to take any question. Thank you very much.
Thank you, Véronique. Any questions in the chat? No? So maybe you can start. Would you now consider that impaired cholesterol clearance as a primary driver rather than a secondary consequence of lymphatic dysfunction based on your data?
But in which setting?
I mean, in general, because you have a problem of cholesterol clearance in both atherosclerosis and in lymphedema. So do you think that this impairment of cholesterol clearance is the primary driver of lymphatic dysfunction, or it's the consequences of?
Yeah, I guess, if you take secondary lymphedema, so one thing that I didn't mention is that unlike in ApoE knockout mice that have, they are hypercholesteremic, the lymphedema patients are not hypercholesteremic. They have normal cholesterol level, but they have lymphatic insufficiency. So we definitely start with this lymphatic insufficiency, and then we see this, accumulation of cholesterol. Unfortunately, in our study, we didn't have access to earlier stage, like really like stage 1, which are the early phase of the disease, to really look at this, and we are looking at that now. So in this case, I think it's really like first you have lymphatic insufficiency, like accumulation of cholesterol, and however, what we found is that this accumulation of cholesterol will lead to or contribute to inflammation. I didn't show here, but it contributes to adipose also remodeling, in particular, and fibrosis, because when we clear this cholesterol, basically we alleviate some of these basically remodeling and changes in the tissue.
Okay. So yeah, based on this, in the context of atherosclerosis, do you think that lowering systemic cholesterol level could have an impact on lymphogenesis and function or not? Or do you think that it's local effects or can be systemic effects?
Yeah. So in atherosclerosis, of course, there is hypercholesteremia, and then this will lead to this cholesterol accumulation. But what we found, and we found that indeed lowering cholesterol will reverse. Like definitely you have a regression of atherosclerosis and improvement of lymphatic vessels, like lymphatic drainage. But what was to us like surprising is like because and people have shown that you have this lymphogenesis, so more growth of lymphatics, but however, to our surprise, this lymphogenesis was not necessarily associated with more or a better lymphatic drainage. So it means like maybe the cholesterol like alter the structure of the vessels and the function, which is what we observe at the time in the skin, but not necessarily in the aorta. So I think it depends. Like, of course, in atherosclerosis, you have hypercholesteremia, so you need to, and it's because you have increased synthesis of cholesterol, so you need to block this. But in lymphedema, we don't necessarily have hypercholesteremia, so we don't need to block the synthesis, and that's why we didn't. I have a lot of questions on this, like why did you use statin or ezetimibe, to reduce the cholesterol in the lymphedema model? But here it's not a problem of more synthesis, but it's really more because you have accumulation of this cholesterol because of lymphatic, drainage impairment. So that's why we decided to use another strategy, which is to clear this cholesterol, solubilize, remove it from the tissue, and see whether we can improve the vessels.
Okay, there are some questions in the chat. I don't know, Véronique, you can see them?
Yes.
Oh, a question from Bernard.
Do you think that the vasorum play a role in the transfer of cholesterol or in correlation with lymphatic vessels? So, in fact, we think that the first part of my talk, when I mentioned about when we look at the reverse cholesterol transport, and in fact this was also shown by Gwen, is that the reverse cholesterol transport is altered by 80%. So when we have impaired lymphatic function, so it means that this pathway is one of the dominant, and surprising, we thought that maybe the venous system could do that, but it looks like the lymphatics is one of the major basically circulatory system that is important for that reverse cholesterol transport. The other question is, do you think that the removal of cholesterol by lymphatic vessels is a direct mechanism or via foam cells? We didn't, the way we assessed that at the time, we use macrophage, basically when we load it with cholesterol, it's just because as a vehicle and to deposit this cholesterol within the tissue, and then to mimic the reverse cholesterol transport, so the cholesterol will be effluxed by the macrophage and then so forth. We didn't look carefully or assess whether it could be via foam cells or not. I think it's quite difficult to do some of these assay with foam cells because they are rare, but it's a good question. I think we still don't know also how the cholesterol alter lymphatic vessels in general, whether is it like a affect the endothelial cells, the smooth muscle cells, overmural cells, and so forth. I think a lot more is needed to understand like how this abnormal cholesterol, particularly accumulating around the lymphatics, whether initial or collecting, affect the function of the vessels. And I think this is a really important aspect that we didn't look at yet.
So if no more question, so thank you again, Véronique. So we'll move to the next presentation. Eba?
Can you hear me?
Yes.
Okay, great. Just waiting for it. It was a fantastic talk, Véronique, really interesting. While this is uploading, I just want to thank Soraya for making this seminar. So I want to talk to you about the lymphatics in the heart. So cardiac lymphatics. So they exist as a superficial plexus in both humans and in mice, spanning the entirety of the surface. And their function, of course, is to drain. And they drain from the apex towards the base of the heart, where are situated the main cardiac lymph nodes. When we look at the different segments, the capillary segments are responsible for the uptake because they're very permeable, so they will let both fluid and macromolecules and other solvents enter very easily. But they also have this active recruitment of immune cells mediated by chemokine gradients secreted by lymphatic endothelial cells. And they also express certain molecules, such as LIV1, which is an adhesion molecule that interacts with hyaluronan or hyaluronic acid. That is glued onto the surface of immune cells that express CD44. So it exists sort of like an immune cell glycocalyx that enables the immune cells that are in the proximity of lymphatic endothelial cells to really adhere and then cross into the lymph to be cleared. If you look further upstream in the segments of the lymphatic tree, similar as in the other organs, we have these pre-collector or collector vessels that are responsible for the transport. They're equipped with valves to prevent the backflow in the system. Different from other tissues, so Véronique was talking a little bit about the lymphatic muscle cells that are responsible for creating the pressure gradients in the heart. It's the cardiomyocytes that do the job. So the rhythmic contractions of the heart is what causes the pulsatile flow in the lymphatic system. Also different from other tissues is that in the heart, these valves that can be visualized here with portocalixtin stain are present not only in the collector segments, but also out in the initial or capillary lymphatics to really ensure that lymph is going unidirectionally and efficiently from the apex towards the base of the heart. Okay, so what happens in cardiovascular diseases? But there's been quite a few studies investigating the changes to cardiac lymphatics in different settings. And for example, in patients with terminal chronic heart failure, whether it's ischemic or dilated cardiomyopathy, you can see changes either in the density and or in the sizes of these lymphatic vessels. So what does these structural changes mean? Well, to get a little bit an idea about the functionality, we turn to experimental models. And again, there's been quite a few different cardiovascular disease settings that have been investigated. And among the one of the things that have emerged from all these studies is that there is a close link between immune cells and lymphatic remodeling. And many of these immune cell populations contribute directly or indirectly to regulation of lymphadenogenic. So some will stimulate it, and others will inhibit it. For example, cardiac infiltration of T cells, activated T cells after myocardial infarction is one of the reasons why you have destruction of cardiac lymphatics. So this can be seen this is experimental models in mice. You can see in the infarct scar after myocardial infarction, there's a destruction and then a regrowth of vessels that look rather aberrant, and also some lymphadenogenic in non-infarcted areas of the heart. We recently showed in the pressure overload model that there was instead a massive expansion of lymphatics. So this just illustrates the fact that depending on the context, you will have different lymphadenogenic responses. In the end, it's not so much the structure that is interesting. When you think about it from a functional point of view, it's really the drainage capacity, like Véronique was also mentioning. So how do we measure this? Well, similar again as Veronique was using in the foot pad, you can inject lymphatic tracers directly into the myocardium, and then you can follow the uptake towards the draining lymph nodes. And directly after myocardial infarction, well, even for 1 month after myocardial infarction, we have very, very little lymphatic transport, so indicating lymphatic transport insufficiency or lymphatic transport dysfunction. And only after around 3 months do we see an increase in the uptake and the drainage towards the lymph nodes, so indicating really something long-term as a lymphatic dysfunction following myocardial infarction. So other people have also used this similar approach and found that there was a reduction of lymphatic transport capacity after pressure overload induced by the transaortic constriction or the TAC, and also in models of HFpEF or heart failure with preserved ejection fraction induced by LNAME and high-fat diet in mice. There was a retention of this lymphatic tracer in the heart, which was not cleared efficiently. So taken together, this suggests that lymphatic dysfunction is something that is shared in different heart failure settings. So what could be the mechanisms? Among the things that can contribute to lymphatic dysfunction is, of course, alteration or reduction of the lymphatic density, also reduction of the capillaries that may become less permeable to fluid or macromolecule uptake, or perhaps not recruiting immune cells as efficiently. We can also imagine valve dysfunction as a way where the system would have more backflow and therefore not be able to transport efficiently. Or we can also have, I think Veronique mentioned this, that there's also increased lymphatic leakage that can occur in the pre-collector or collector segments. And this has particularly been described in metabolic disorders, so in cholesterolemia, but also in diabetes and in obesity. So these are mechanisms. So if we look at these different items, which ones may be involved in heart failure? To explore this, we take use of a previously found difference in lymphangiogenetic capacity between BALB/C and C57 strain. So after TAC surgery, we have this massive expansion I showed you before of the lymphatics in this model, whereas in the C57 strain, we have much more moderate lymphatic expansion. And so you'd argue or expect from these data that fluid drainage would be better in this mouse, and also maybe immune cell clearance would be better in the BALB/C after TAC. But this is not what we see. We actually see similar levels of myocardial water content in both strains, so indicating that this expanded lymphatic network is not serving its purpose. Similarly, when we look at immune cells, the levels are actually quite comparable between the strains. So again, indicating that lymphatics are probably not doing their job correctly. When we look at the immune signatures between these different strains, they're not exactly the same. Obviously, there's different immune settings. We have a lot more of IL-1 beta and IL-6 in the BALB/C strain, and maybe more TNF-alpha in the C57 strain. And these differences in immune signature is also associated with differences in the expression level in the heart of the lymphangiogenetic driver. So this increase in VEGF-C, specifically in the BALB/C, is associated or can explain perhaps this really expansion of the lymphatic vasculature in one strain and not the other. So if we look at as mechanisms of lymphatic dysfunction, it's pretty obvious that at least in the post-TAC, in the post-TAC setting, is not a reduction of lymphatic density that explains it. So what about the capillary uptake capacity? So to address the uptake capacity, we did a single-cell RNA-seq analysis, focusing in on the LEC clusters, or lymphatic endothelial cells, and we saw lots of upregulated and downregulated genes. And then when we analyzed the subpopulations that made up all the lymphatic endothelial cells in the heart, we could find that there was a clear expansion of this blue cluster here, which had the capillary profile. So this agrees with what we see in immunohistochemistry, that there's a selective expansion of capillary lymphatic endothelial cells after pressure overload. So then we looked at the capacity of these capillary lymphatic endothelial cells to interact with immune cells. So different from our expectations is we saw inflammation lingering in these hearts. We thought that there might be less of the chemokines that drive this attraction of the immune cells towards the lymphatics. We instead saw an increase both at the RNA level and at the protein level of CCL21, which is one of the major chemokines that drive immune cell attraction. So you can appreciate it here in red, CCL21. Similarly, we also saw an upregulation of this adhesion molecule that enables the immune cells to interact with lymphatics, increased at the gene level and also at the protein level. It's here in blue. So really no reason for the immune cells to ignore lymphatics. They should be pretty visible and pretty attractive, and still inflammation is chronic in this model. So clearly not any reduction of capillary uptake. I didn't show the data, but we also investigated endothelial cell junctions, and we did not see any major switch in these specialized button junctions that allow the lymphatic endothelial cells to be permeable to fluid. So for us, there's no structural molecular evidence that capillary uptake capacity is reduced. But what about valves? So as I mentioned before, you have to use either injection of dyes or immunohistochemistry, for instance, with podocalixin, that you can visualize these valves present in the capillary lymphatics as well as in the pre-collector lymphatics. In addition, our data from the single cell analysis show that this LEC3 population, which was carrying signatures of lymphatic endothelial valve cells, was really reduced in the following pressure overload in BALB/c mice. So this gave an indication that perhaps something's going on with the valves. And so we quantified the number of valves, and as you can perhaps appreciate in these images, that there were whole segments following TAC surgery that were devoid of valves. Mostly the capillary segments actually were devoid of valves, and when we quantify that, we saw a clear reduction. So this could, of course, be either a dropout of valves or just insufficient valvulogenesis. But as a whole, it's pretty obvious that if you have a problem with the valves, you will have more backflow in the system, and that could definitely be one of the mechanisms that contribute to lymphatic dysfunction in the heart failure setting. What about increased lymphatic leakage in these pre-collector segments? Again, looking at the data from the single cell analysis, but now in the pre-collector lymphatics, we know that these cells depend on a very specialized structure that are more like tight junctions, zipper junctions that are made up of one of the molecules is claudin-5. And we could see that there was actually a reduction after TAC of claudin-5. And this again could be confirmed at the protein level, where you see the lymphatic endothelial cells in gray, and that they lack this claudin-5 in red. So indicating that something in pressure overload condition is causing the loss of claudin-5. We wanted to know a little bit about the molecular mechanisms that might drive this loss of claudin-5 during pressure overload in BALB/c mice. And so we turned to an in vitro analysis, and we saw that stimulation with IL-1 beta, which is one of the cytokines that is highly upregulated in the setting of TAC in BALB/c mice, was sufficient to drive this downregulation of claudin-5. And when we look at the cell monolayers, you can, I hope, appreciate that there are like holes in this monolayer. So this would indicate that there might be some leakage going on, notably in the pre-collectors, and that could certainly cause an increase in lymphatic leakage, and so that could also contribute to lymphatic dysfunction. So to conclude, what I've shown you is that depending on the cardiovascular disease context, stimulation of lymphadengenesis doesn't look the same. More importantly, however, that it seems to be a shared feature in many different cardiovascular diseases that the lymphatics dysfunction. And when lymphatic transport is not sufficient, well, obviously it can contribute to both chronic edema and inflammation, and as Veronique suggested, also to build up of cholesterol, although this is something we have not addressed. While in the TAC model in BALB/c mice, we did see an upregulation of immune attraction pathways, indicating that this is some sort of maybe compensation that the lymphatics are trying to do. The loss of valves that we observed and the potential changes in lymphatic pre-collector barrier could definitely contribute to the dysfunction. I haven't shown you the data, but in other models, for instance, after myocardial infarction or post-TAC, but in another strain, C57, we do see this loss of button junction. So perhaps the mechanisms underlying the lymphatic dysfunction are different, but overall it's a shared feature that lymphatic transport capacity is reduced. So if we look at the perspectives or the the work that still remains to be done. Obviously, we're very interested in anything that can restore lymphatic density, but I think also that it's really the time to consider what type of therapies we could use to restore lymphatic function. Another thing that I really see as a major outstanding question is whether or not this remodeling that we know occurs in humans with cardiovascular diseases. So the changes in lymphatic structure, is that associated with lymphatic dysfunction? And actually, there's absolutely no data on that because it's such an invasive procedure. And the only data that exists in larger mammals is from pigs, where they injected into the myocardium gadolinium and then followed the transport up to the draining lymph nodes. And so this is really, I don't know how we will address this, but it's certainly one thing that would need to be addressed to be able to propose or not therapies in the future. The last thing I want to mention is that there was a very large prospective cohort analysis of the coronary artery disease patients, and they found that very low levels of plasma VEGF-C was associated with worse outcome. So indicating that, maybe it's not the cardiac lymphatics only that will benefit from high VEGF-C, maybe it's systemically, but definitely I think further investigating the role of lymphatics in cardiovascular diseases is most intriguing. With that, I want to thank the people that contributed to the work, my former PhD students, Caroline, Chloé, and current PhD students, Oceane and Emily, and our international collaborators, and of course you, for your attention. Thank you.
Thank you very much, Eba, for this insightful presentation and clear presentation. So any question in the chat? So maybe I can start if there are no questions.
Maybe.
Oh, you have a question here?
Yeah,
can
just,
Eba?
Yeah.
Ah,
sorry about that. Yeah. Maybe just a quick one, like very nice and beautiful pictures, Eba. Like, I was just wondering the difference you see between the black 6 and the biopsy. Do you think it could be also related to T cells in particular? Because, we know that, the black 6 usually are more like promoting a Th1 kind of response, like the other one Th2, like, whether you look at a bit more specifically of the T cells. I know you've shown some like changes in maybe some inflammatory cytokines, but a little bit more on immune cells, since they're also known to regulate lymphogenesis.
No, it's a very relevant question. Thank you. But so what we saw was more T cells in the biopsy, actually. That was a bit surprising. We haven't really analyzed the activation states and so on in the T cells in one model versus the other. I think also it should be noted that we really used female mice. And so if you use the C57 male mice, you get quite a different response. You get more T cells in the males than actually in the females. But yeah, definitely a very relevant question. Thank you. I saw a question in the chat. To what extent do you anticipate that the lymphatics can be repaired without taking into account the non-functional fibrotic scar that replaces the cardiomyocytes? Okay, so different VEGF-C delivery therapies have been able to restore fluid balance more rapidly. So I guess that is one way to say that it is possible with VEGF-C, for instance, to accelerate the endogenous lymphadenogenic response. Does that benefit the scar? I don't think so, because the scar obviously is devoid of contractile elements, and so you don't really get much functionality out of lymphatic vessels that don't have any contractility. I don't know if that answers the questions, but thank you.
So maybe related to this question, how reversible are the lymphatic alteration once cardiac stress is removed? Do you think that it's something that can be reversible or not?
I personally believe that it's the inflammatory environment that messes up the lymphatic function. I think also maybe one thing that I didn't mention in the talk that is probably relevant, at least in some settings, is the fact that the lymphatics depend on cardiomyocyte contractility. So any heart failure condition is likely to indirectly reduce the capacity of its lymphatics to transport. So is it reversible? If you can treat the heart failure, then you can treat the lymphatic dysfunction.
Okay. So if there are no more questions, thank you again, Eba. And we will move to the last presentation. Seppo, if you can share your presentation.
So thank you for invitation to talk about lymphatics, which we have been working quite some time. I will first show some stuff about lymphatics and atherol, and then a little bit about our clinical trial with lymphatic growth factor. We've been working with lymphatics already more than 15 years, and I'm not going too much in this old data, but as you can see, there is a lot of lymphatic vessels in, for example, coronary atherosclerotic lesions. And that then brought us to mouse studies, which we've been using 3 different mouse models. One is K14 promoter-driven soluble VEGF receptor 3 decoy. K14 is expressed in keratinocytes, so this mouse produces decoy VEGF receptor 3 in the skin. Then we have VEGF-D knockout mouse. We can't have VEGF-C knockout because that's embryonic lethal. And then we have this Chi mice that was also presented in Veronica's presentation. All these have been crossbred with LDL receptor ApoB100 only mice, which I think is the best background for the athero studies because its lipoprotein profile is more like human. And just to, if you are not fully familiar with the VEGF receptor family, so lymphatic receptor is this VEGF receptor 3, and that's the decoy we are using. And it has 2 ligands, VEGF-C and D, which then, when they are proteolytically activated, can also bind to the VEGF receptor 2, that is angiogenic receptor. And the first picture is just to double check whether something happens to lymphatic vessels in these mice, and these are atherosclerotic lesions. From here in red circle this soluble VEGF receptor 3 mouse, and then Chi mouse. And the white bars are adventitial blood vessels, and then gray bars are the lymphatics with live 1 immunostaining. So you can see that both models really lack lymphatics in the atherosclerotic lesions, and here is also lectin, CD31, and then poroplanin staining that verifies that really these athero lesions, which in control LDL ApoB mice have nice lymphatics, these are missing from these animals. And the first surprise to us was that actually blood lipid levels go sky high. This is a soluble receptor 3 mouse, and you can see that even with 2-3 weeks with lipid Western diet, we have something like 50 millimoles of cholesterol, which you can get nowhere near with most other models. And also triglycerides go very high. What happens is actually that in this decoy receptor mouse, the total cholesterol is more and more carried in LDL fraction, whereas in normal mice, of course, you have most of the cholesterol in HDL. And that mouse then was used for many, many studies. We looked lipid absorption, LDL turnover, glucose tolerance test, and so on. All those were normal. So blocking the lymphatic receptor didn't affect those. But what happens, what you can expect is that these animals had increased atherosclerosis. We had young mice and then middle-aged mice, if you wish. And here in white bar is the decoy receptor mouse. And you can see that both in young and then older animals, they have clearly more atherosclerotic lesions. The gray bar is this Chi mice that also has increased atherosclerosis. So this model told us that when you mess around with the VEGF-C and D signaling, you will, and you have a LDL receptor background mouse model, you will get increased atherosclerosis. This is how the lesions look like, so pretty mature type lesions at 6 weeks with both macrophages, cholesterol crystals, and smooth muscle. in the developing lesions. Well, then we thought that this is as we expect, and really because we've been mostly working with VEGF-D, we took the VEGF-D knockout mouse and did the same studies, and these mice had even more cholesterol in their blood in 2 to 3 weeks, so 60-70 millimoles, and the blood looks really milky, so like chylomicronemia in humans. What we then saw in these animals was that lipoprotein profiles were totally distorted, so that in black bars here, VEGF knockout mouse has a lot of VLDL and actually large chylomicron lipoproteins as compared to the background mouse. The lipoprotein profile distribution in parties also was pretty abnormal. So when you knock out VEGF-D, you will get this type of profile. There were no changes in lipoprotein lipase or hepatic lipase, which were the first suspects that would cause this. There were no real differences in LDL turnover with radiolabeled ligand. And there were no changes in lacteals in the intestine, because you would think that maybe the VEGF-D causes something in the lacteals, which are important for lipid absorption, but that was not the case. So then when we looked further with lipid absorption with triolein, that was normal. But what was abnormal was actually that the chylomicron remnant uptake to the liver was significantly reduced. And that means that here in black bar, so that chylomicrons, which we saw in the plasma with the very high cholesterol levels, kept circulating. And the situation was pretty similar to human type 1 hyperlipidemia. So we were quite excited that, hey, what might be happening here? We did a lot of RNA sequencing, and there are a lot of changes in, for example, downregulation in lipid metabolic genes and so on, and upregulation in some of the ERK signaling pathways. I'm not going into those, but I'm showing you this one, which is syndecan, that was very much downregulated. And syndecan is actually the most important receptor for chylomicrons or chylomicron remnants in the liver in the absence of LDL receptor. And what we saw here in black bars, that syndecan and also its neighboring helping receptor were clearly downregulated, and also that happened in the protein level. So we could, and also the immunostaining syndecan controls and knockout mice clearly reduced. So we were able to come up with the mechanism that VEGF-D knockout somehow produced situation that syndecan receptor was really downregulated in the liver, and that allowed chylomicrons to circulate much longer. However, surprisingly, these especially young VEGF-D knockout mice had much less atherosclerosis than you will see the control in white bar and this VEGF-D knockout in black. So clearly the young mice, these are up to 2 months old, were really almost protected from atherosclerosis. Then when you go to the 1-year-old mice, the situation seems to level off. But there was clearly a surprising finding that regardless of 60-70 millimoles of cholesterol, these animals didn't have very much atherosclerosis. And actually, then when those of us who are old enough to remember Silversmith and his studies, actually seems to be the situation that these chylomicrons are way too large and they can't enter subendothelial space. And there is a lot of old literature about that. And the situation is very similar to human type 1 hyperlipidemia, so that these patients also do not have increased atherol in spite of very high plasma lipid levels. So in these situations, these lymphatic growth factors seem to affect liver and lipoprotein metabolism. We also then went into the infarction studies in mouse heart, in this case, this decoy receptor, and used LAD ligation model to see what happens. As we saw already in previous presentations, there is nice lymphatic network in the heart muscle. But when you have this soluble decoy receptor, the lymphatic network is distorted. It is there. And actually, as we heard, there is slightly increased amount of cardiac lymphatics in this type of decoy receptor model. When we did the infarction, so it looked like that infarction scars were a little bit larger in these animals. And indeed, that was the case. So if you look infarction scar size, they were almost double as compared to the control animals. And this led to the survival deficit, so that here you can see that the soluble VEGF receptor mice really didn't survive, nowhere near as good as the control animals did in the case. So from mouse studies, I would conclude that all these models show cholesterol levels that are very high already after 2-3 weeks lipid diet. And this soluble VEGF receptor 3 mouse, which this system blocks both VEGF-C and D effects, they have accelerated atherosclerosis together with the very high lipid levels. But when you only take off VEGF-D, in spite of very high cholesterol levels, they do not have excessive atherosclerosis. And then these soluble decoy mice also had a higher mortality after MI and a little bit higher scars. So actually, VEGF-D knockout mice have working lymphatic system, not perfectly working, but it is there because VEGF-C growth factor is present. So then these have been all basic studies that have led us to the clinical use of VEGF-D, because we believe that both angiogenic and lymphangiogenic growth factor might be beneficial for heart. And so we started already 10 years ago for refractory angina trial. Refractory angina refers to patients that suffer from very severe angina despite optimal medical therapy, and they are not suitable for any revascularization therapies. And number of patients is rapidly increasing because of the aging population. Some 30,000 new cases per year in US and Europe. In these patients, we believe that, as you well know from the clinics, that collateral arteries, which you will show here, can rescue the heart. Here is circumflex coronary artery that is blocked, but then the distal part is filling because of through the collaterals. And we believe that therapeutic vascular growth could have similar beneficial effects in man. And just to double check this VEGF receptor signaling picture, so we use here VEGF-D delta and delta C, which is a proteolytically activated form of this full-length VEGF-D. And this proteolytically activated form is likened both receptor 3 and receptor 2. So it's both angiogenic and lymphangiogenic. We've done a lot of studies signaling whatever with this VEGF-D molecule. And I can say that this is a combination of therapeutic angiogenesis and lymphangiogenics, and it's a new approach for cardiac gene therapy. And the benefits, what I believe are important in human context, is that the VEGF-D signaling kinetics is much less aggressive and lasts much longer than VEGF-A, which actually shoots up in 2-3 hours and then disappears very quickly and causes a lot of, for example, leakiness. So VEGF-D shows no or very little edema, and therefore the risk of cardiac tamponation is very low. VEGF-D diffuses very well in myocardial extracellular tissue because it doesn't bind to heparan sulfate proteoglycans. And it also binds to neuropilins, which are the co-receptors for VEGF receptor for VEGF effects, but it doesn't bind to VEGF receptor 1, which is chemotactic for monocyte macrophages. So it should reduce a little bit the inflammatory risk what we have. We use targeted delivery with a catheter called NOCA catheter. So you put it in iliac artery and then into the cardiac chamber, left chamber. And the tip of this catheter measures electric activity. So purple here is normal. So this part of myocardium produces EGG and is healthy. Red means no activity. So here you will see infarction scar. And around that is the hyponating myocardium in blue and green, which is alive, but very sensitive to arrhythmias. And if worse comes to worse, this scar just keeps expanding. And then once you have mapped the heart and located the ischemic and then the hyponating myocardium, you can push out a tiny needle. And these brown spots here show the injection site. So we do not inject in the scar, of course, because it doesn't produce anything, but around the hyponating myocardium. And in our clinical trial, we have gone one step further, and we have added actually radio water PET scanning. Radio water gives you quantitative blood flow in the myocardium. And now here is an example of a patient. So here is from the basis of the heart, purple area that is alive and produces EGG. But then this catheter also actually measures the wall movement. And here you will see this red island that doesn't move. So it's purple, it's alive, but it doesn't contribute to heart function. So it's the hyponating myocardium. And on top of that, we will put the perfusion map, and you can see that here red is good perfusion, yellow, green is bad perfusion. You have here a island of poor perfusion that doesn't contribute to the heart function, but it's alive. And here are the injection sites. You will see how you then try to target your therapy as perfectly as possible, I think, in living human heart. And we've done already phase 1/2A study that's been published. And actually, those of you use adenovirus as a delivery. Those of you who work with viruses realize that this is very low dose, 1 × 10 to 11 viral particles. So because of the very targeted injections, the volumes are low. And that's why the inflammatory properties that adenovirus might cause are much less likely to happen. In the phase 1 trial, we saw clearly in radio water perfusion assays. Here is 1 treated patient. Here are the purple treated areas. Baseline perfusion is shown here. So red is good perfusion, red and yellow. And 3 months and 1 year after the treatment, you can clearly see that the red perfusion and yellow has expanded significantly. Another treated patient shows here the baseline. And then 3 and 1 year after treatment, clearly improved perfusion, whereas randomized controls shown here do not show any improvement. And actually, the myocardial perfusion reserve was significantly improved in this material after 1 year of the treatment. So we now have actually 8 years follow-up from this phase 1/2A study. And surprisingly, we saw that Canadian Cardiovascular Society's score for angina symptoms was significantly improved in these long-term follow-up patients. Of course, this is only anecdotal and hypothesis generating, but was quite interesting. And also 8 years, we didn't lose but 1 patient out of 30, which were in this phase 1 study, which tells you that actually these refractory angina patients, their mortality is very, very low, but quality of life is very bad. So a lot of people contributed here. My group our hospital people. I'm just thanking our study team for the clinical trial, Jens Kastrup in Copenhagen, Anthony Matthew in London, Marin Grönkels in Vienna, and Sam Chimikas at UCSD. And the vector was made by Finvector company. So thank you very much.
Thank you, Seppo. Are there questions in the chat? Maybe I can start. So in terms of therapeutic, which patient population do you think would benefit the most from lymphatic targeted therapies? Like patients with MI or FPF or pressure overload cardiomyopathy?
Well, this is very difficult to say yet, but what we know is that in terms of refractory angina, the most common pathway to refractory angina is that you have coronary heart disease, not any cardiomyopathy or anything like that, and that then you have had 1 or 2 stenting PTCA operations and bypass surgery, and still that doesn't help. And there is an argument that there must be a lot of microvascular abnormalities in this type of refractory angina situation, so they are under optimal medication. We are looking for several biomarkers that could explain or target the treatment, but at the moment it's a severe atherosclerosis and coronary heart disease that seems to be the major population. I didn't show you the patient details, but the cases and controls were very nicely balanced regarding like high cholesterol, hypertension, type 2 diabetes, and so on. So we can't fully answer yet that, but athero patients are the most likely to benefit.
Thank you. So there is a question from Eva. Could you see the question, or I can tell you? How do you interpret the finding that LPA levels correlated with VEGF-D benefits? More benefit of VEGF-D in patients with more hypercholesterolemia or dyslipidemia?
Yeah, we measured for sure. Sam Chimikas has measured Lp(a) also from these patients, and the same finding that we had in phase 1/2a study came through here, that the highest tertile or Lp(a) benefit patients seem to benefit most. I think one argument is that Lp(a) is actually a little bit anti-angiogenic, so it could contribute to the microvascular dysfunction in the myocardium, but we don't know fully yet whether that's cause or consequence. But Lp(a) is a potential biomarker, which was also measured in this phase 2 trial.
Another question. Since the therapy was a combined effect of angiogenesis and lymphogenesis, what could be the contribution of angiogenesis compared to angiogenesis? Did the patient have lymphogeography?
We didn't have any lymphographies, but I think that one of the main reasons that we believe that VEGF-D might have more beneficial properties than, for example, classical VEGF-A 165 is that there is always some leakiness in the whenever you induce angiogenesis, even with VEGF-D. And now when you improve the lymphatic function, which we have shown in our big model with dextran, that has been injected in the apex of the heart, and you can clearly see the improved drainage of the through the lymphatic channels. So I think that it reduces the minimal edema that there must be in the myocardium, and that and reduces the strain, and therefore seems to be helpful. Also, I didn't go through the biology of HFD, but it's also stimulating progenitor cells to some extent. So there might be some regenerative aspects here as well.
Thank you. No more questions in the chat. So with this, we will conclude this webinar. Thank you again to the speakers, and thank you all for attending. And I look forward to seeing you at the NSFA Congress in Biarritz on June 24th. Goodbye.